Okay, so this is video 2. We're going to start talking about translation, which is going to be the process of taking our piece of messenger RNA and turning it into a protein that can then do something within cells, whatever that protein is supposed to do. We already talked about the word codon. So a codon was just 3 mRNA nucleotides. So if I look at my picture here, kind of jumping ahead. So 1, 2, 3. So this is a codon, a series of 3 messenger RNA nucleotides. And then so an anticodon is going to be a series of 3 transfer RNA nucleotides that's going to be complementary to the mRNA codon. I'm not going to show you that just yet, though. You'll see that in a minute. And we're going to talk about amino acids and proteins later. I do want you to know that there are 20 amino acids and 64 different codon combinations. I'm only asking you to know 4 of the 64, so I feel like that's pretty doable. The first one is the RNA sequence AUG, so it's adenine, uracil, guanine. That one is significant because it's the start codon. It starts translation, and it also codes for an amino acid called methionine. I want you to know that too. We won't really use that too much, but at least this way you know one of the 20 amino acids. The other 3 codons I want you to know are UAA, or uracil, adenine, UAG, and UGA, and those are the stop codons. They just signal the stop of translation. So during translation, when one of those sequences is reached, it stops. And those don't code for an amino acid. They just stop translation. Down here, so there's 4 steps in translation, says what they are. Again, I'm not going to necessarily read you every word. We passed out the notes before the weekend. So there was 3 of them, and the 4th one, which I didn't have room for. So initiation, elongation, translocation, and termination. Make sure you know what those things are. Keeping going, protein synthesis is just making proteins. The word synthesis, can be used in non-science settings. And then gene expression is what transcription and translation together are doing. So transcription gave us the messenger RNA from the DNA, and then translation is going to take that information in the mRNA and give us a protein. So it is the expression of the gene in the DNA, or it's the gene being expressed or activated, or whatever word you want to use there. And I kind of already said those things. We'll see those in my drawing, and that'll take us to here. So again, I'm not going to stay on this slide too long. I want you to, you can pause the video and read these things, but I think we already said most of these. Polypeptide we haven't said in a while. Remember, that's basically like, think of it as like part of or a piece of a protein. And that might not be the best definition for that, but it's good enough for us. And so make sure you know those words, what those things are, definitions, that sort of thing. And so we're on to my video here. Our video. Sorry, this is the video drawing. So anyways, our piece of mRNA, which you saw in the last video, created from our DNA, has left the nucleus and found a ribosome. So this is just my world's worst ribosome drawing. Within that ribosome, the piece of mRNA, so this is my mRNA, this ribosome is going to take transfer RNAs, tRNAs, that are going to be complementary to the codon sequence. So the end 3, that's kind of misleading. The end 3 nucleotides. So that is going to be an anticodon. Series of 3 tRNA nucleotides that is complementary to our codon. So again, this is still RNA. This is tRNA. So what's going to be complementary with an adenine? Uracil. with a guanine would be cytosine, and with a cytosine, guanine. So on the other end of that tRNA, there is an amino acid. I'm just going to call it binding site because I don't know what else to call it. And there's going to be a specific amino acid that's going to bond to just this one. So the next tRNA is going to have a different binding site and a different amino acid is going to attach there. So this sequence, I kind of did it in like reverse. So our first codon would be CGA because it's going to get read this way. So I was committed to this drawing by the time I thought of this. I kind of drew it like mirror image. But CGA is going to code for an amino acid, which I'm just going to represent as a ball. And if you guys would look up, there's a chart that has all these things that you can look up what amino acid it is. And like I said, I don't care that you know this, but CGA, the codon would be arginine, which is just one of those 20 amino acids. So again, that is a type of amino acid. And so I can't change this because my pen drawing, but that piece of mRNA is going to then work through the ribosome, or you could think that the ribosome shifts down. That's the translocation step. So then imagine that my ribosome is now covering this next codon, which would be GUC again, because I kind of did it backwards. And so we're going to have a new tRNA with complementary nitrogen base pairing. So this would be a C and a and a G. And it's going to bond with a different amino acid. So if it is GUC, it is the amino acid valine. And then once that's done, these two amino acids are going to get linked together. And for the sake of me not spending a ton of time doing this, we would then do the next one with a new tRNA, complementary base pairing A, C, U, and I don't have, oh, I can't get another color, and another amino acid. And so if it's AGU, it would be amino acid serine. And then you can start to see how these amino acids are going to get linked together. And a chain of amino acids we could call a polypeptide, or if it was a really long chain of amino acids or several polypeptides stuck together, that is how we make a protein. So to kind of recap the whole thing, so both these steps would be called gene expression. And that's transcription plus translation. So we start with DNA. There's a piece of DNA that's to be transcribed because we need to make a protein to do something within our cells. It starts at the promoter region. We make this piece of messenger RNA, and it's messenger RNA because we're going to make a protein out of it. RNA polymerase creates this piece of RNA with complementary base pairing rules using uracils instead of thymines because it's messenger RNA. Our piece of RNA then leaves the nucleus, finds a ribosome. That ribosome finds complementary tRNAs with anticodons that are complementary base pairing rules to our codons. And the binding site of that tRNA is going to take a specific amino acid and create a protein. So you can see that the RNA came from our DNA, and which specific amino acids are used is dependent directly upon that RNA. So the DNA in your cells is the instructions for the proteins that your cells create.
That is just really cool.